Okay, this is the identification of my unknown bacteria. So I collected my specimen on January 16th, 2020 at approximately 12:45. I obtained this bacteria from the light switch that is by the closet in the microbiology lab. The plate was then incubated until the next class on January 21st, 2020. My goal was to test the bacteria that is on the light switch in the microbiology lab. I continued to achieve this goal by using different staining techniques to identify the bacteria, and I expected to see gram-negative bacteria from the beginning. I also expected to see similar bacteria to the bacteria my classmates were getting from the room, just because it's surfaces that are all commonly touched by the same people. Background information, I concluded that my bacteria was gram-negative because of its pinkish red color. After being incubated, my bacteria was also a form of spirilla because of its spiral nature, and they are not in any sort of pattern. My procedure, I perform the following techniques to identify my bacteria: aseptic technique, isolation, hanging drop method, simple staining, gram staining, acid-fast staining, endospore staining, capsule staining, negative staining, blood agar, oxygen and growth catalyst, and oxidase. However, there was supposed to be more labs that were mannitol, salt agar, MacOnky agar, eosin methylene blue agar, triple sugar iron agar, IMVIC antimicrobial susceptibility testing, or the Kirby-Bauer method, but I couldn't perform these because we obviously had to leave campus. Okay, so first, the most important thing when collecting your bacteria and doing these labs is aseptic technique. So to ensure that my bacteria was not contaminated, I always made sure that I wiped off my work area with a disinfectant and start my Bunsen burner. I first started out by smearing the bacteria that I collected from the light switch on my agar plate. In order to collect my bacteria from light switches without contaminating it with bacteria from the air, I didn't take the cotton swab out of the package until I was right next to the light switch, just to stop the stuff in the air from contaminating my cotton swab. I made sure that I stayed near the flame because this prevents my bacteria from getting contaminated with different bacteria. My smear plate was inoculated and my results were not the best. There was a few single colonies, but it could have been better. And I don't have a picture of my smear plate, but here is an example of a good smear plate where there's many single colonies, which makes it easier to take those single colonies and put them on different agar plates. The isolation of my bacteria and the hanging drop method. In order to better isolate my colonies, I created an isolation streak plate. A streak plate is a widely used method to separate individual bacteria from mixtures. The goal of this is to have fewer and fewer bacteria as you move around the plate, aka you want more single colonies to make it easier to move them to different agar plates. My plate was not successful. I had the general pattern shape down, but I used too much bacteria and only had a few single colonies. However, I did do this a few times and I eventually got one that was approved. For the hanging drop method, a drop of fluid containing bacteria hangs under a cover slip in the well of a depression slide. I successfully performed this technique and could see my cells. However, because of the lack of color in the cells and the hanging drop method, the bacteria was very hard to see and focus on. Simple staining and gram staining. Simple staining uses basic dyes such as methylene blue or safranin. The background is stained, not the actual bacterial cells. I successfully performed the simple staining technique. I determined that the shape of my bacteria is spirilla because of its sporadic and spiral nature. And here's a drawing of what my bacteria looked like under the microscope, under 100x. And then the gram staining technique differentiates by color. The technique starts with the primary stain, crystal violet, when it is applied. Next, that is rinsed off with water, and the slide is now covered in grams iodine. Next, the decolorizer acetone alcohol is used to rinse the grams iodine off. Lastly, the counterstain is applied and rinsed off with water. The two major groups of bacteria that you will see are the gram-positive and gram-negative bacteria. Gram-positive cells will appear purple and gram-negative cells will appear pink. My bacteria ended up being mostly gram-negative because they were purple. So I was right about that in the beginning, that it was going to be gram-negative, because you had said that it was pretty common to get gram-negative bacteria. And here's a drawing of what my slide looked like under the microscope on 100x. Capsule staining and negative staining. So capsule staining techniques stain the bacterial cell and background and leave the capsule intact and unstained. The capsules are composed of polysaccharides and or polypeptides. I have no picture of my capsule stain slide, but this is what a capsule stain slide should look like. Negative staining is an indirect simple stain that uses dyes that have an acidic chromogen that is repelled by the bacterial cell. Just like all simple stains, the background is dyed, but not the bacterial cells. I have no picture of my negative stain as well, but I successfully used this technique. This is what a negative stain slide should look like. So for acid-fast staining, it is a differential staining technique that differentiates between acid-fast and non-acid-fast bacteria. Acid-fast cells have waxy cell walls and contain mycolic acid. The mycolic acid makes these organisms resistant to desiccation and disinfectants. The waxy cell wall also makes it very hard for these organisms to be stained. I successfully performed this technique, and my results ended up blue with the spiral cells sporadically arranged. And here's a picture from my slide under the microscope. Endospore staining. So endospore staining is classified as a differential stain where heat-fixed smears are steamed over boiling water with the primary stain, malachite green. Heating the endospores with the primary stain allows the stain to penetrate and dye the spore coat. The slide is then cooled, and as the spore cools, the stain is trapped inside. Smears are decolorized and then counterstained with a secondary stain, which is safranin, to dye the vegetative cells. I successfully performed this staining technique, and my cells ended up being a mix of pink and blue cells. Like, it was pretty equal. And here's a picture of that under the microscope. And blood agar was, I think it was the last lab that we did in person. And blood agar is a medium that is both enriched and differential. The iron and other extra nutrients are needed by fastidious streptococci and a number of other pathogenic bacteria in order to grow. Some bacteria grow on it, but some simply break down the blood cells embedded in the agar. Bacteria is alpha hemolysis if it has green color to it caused by a breakdown of hemoglobin. The bacteria will appear to have a halo or partial clearing around the bacteria. Beta hemolysins cause complete lysis of red blood cells and breakdown of hemoglobin. This leads to the colorless clearing of the blood agar around the colony. The blood is completely digested. All that is left is agar. If the organism has no hemolysis, it is reported as gamma hemolysis. There will be no change in the appearance of bacteria. My results ended up having a colorless clearing around the colonies of bacteria, which you can see here, like the clear area around where I put my bacteria. And so because of this, I concluded that my bacteria is beta hemolysis. As I mentioned over here, the colorless clearing of the blood agar indicates that it is beta hemolysis. The oxygen and growth lab. So this test determines if an organism produces catalyst by lacing an inoculum on a glass slide. A few drops of H2O2 is then placed onto the cells. If the organism produces catalyst, there will be bubbling that appears due to the production of the O2 gas from the breakdown of the H2O2, and the organism is considered catalase positive if this happens. This test is extremely useful in distinguishing gram-positive cocci from another. So Staphylococcus and Micrococcus are catalase positive, can be differentiated from Enterococcus and Streptococcus, which are catalase negative. So I was able to successfully perform this technique. My bacteria ended up being a catalase negative because of a lack of reaction. This means my bacteria could very well be Enterococcus or Streptococcus. So I was wrong about the blood agar being the last one we did in person, because I did this one in person. I just don't have a picture of it. So oxidase and results. This technique is used to determine if a bacterium or interest has cytochrome C oxidase in its electron transport chain. To test for the presence of this enzyme, a reagent called oxidase reagent is added to the cells of interest. The reagent is a redox indicator that is colorless when reduced and dark blue or purple when oxidized. The oxidized reagent will then donate electrons to cytochrome C oxidase if it is present. By donating these electrons, the reagent is then oxidized and it will turn purple. And the purple indicates that the organism produces cytochrome C oxidase. And I was also able to successfully perform this technique, and my results did not have a reaction. There was no reaction, so it was negative, oxidase negative. And I don't have a picture of this one as well. So this is the first one that we could not do in person, so we did this online. So mannitol salt agar is an example of a medium that is both selective and differential. This medium contains a high concentration of salt, which makes the medium selective by inhibiting the growth of most bacteria, but allowing salt-tolerant staphylococci to grow. This medium also contains the sugar mannitol and pH indicator phenol red. Disease-causing staphylococci ferment the sugar mannitol. Mannitol fermenters release acid into the medium, which turns the phenol red in the medium around the colony yellow